Very warm welcome again. And the talk that I'm giving here is Scalable Metabolomics in Population Health. And my name is Bijon Chatterji. As you have seen already, I'm globally responsible for population health here at biocrates, and particularly also for biobanking topics. And in this talk, I would like to explore a bit together with you how metabolomics transforms population-based studies through scalable, decentralized, and standardized approaches. And did you actually know that metabolomics is the only omics discipline directly linked to the phenotype? So kind of making it indispensable for understanding health and disease. I would say let's take a closer look at that and see what we got. Briefly, we have three major topics that I would like to cover here. Is first of all the global perspective, highlighting how biocrates consistently sets new standards to address analytical changes in this field, and I would also outline the key requirements for population studies and demonstrate the innovative solutions that we can offer to make to meet these needs. So you see here the global slide for large cohorts and biobanks worldwide. And in the last decade, global population studies and biobank initiatives have collected millions of samples. But you might agree that, and we have seen it in the last two presentations, analyzing these cohorts at scale remains a challenge. And most of these studies were driven by genome-wide association studies. You're pretty much aware of the term GWAS. And as mentioned before, the genome would tell you what could happen. But it is actually the metabolome that would tell you what really happens. So in other words, it is the functional readout of the genetic information. And for large cohorts, for large cohorts, scalable metabolomics is the solution, and it provides actionable insights into pathways and disease mechanisms. And we from biocrates, nowadays lead this field. We enable researchers to tackle challenges in population health with technologies that are not only robust, but also globally harmonized. And this brings me to the next slide, and what I've mentioned, setting global standards, which is crucial in metabolomics to ensure that data from different laboratories can be compared and pooled. And the kit technology that we have developed are actually rigorously tested in inter-laboratory trials. I'm going to outline that a bit later to guarantee this. You should be also aware that there are this is the most cited metabolomics technology worldwide, with more than 1,200 peer-reviewed publications, and shows you how many cohort studies around the world, as you can see on the screen, rely on our technologies. Nowadays, not only North American and the European cohorts, where we've seen historically a lot of, and Tanya Zeller has demonstrated that. But in the second talk, we see that we have a rising number of biobanking initiatives and large cohorts from Asia. So it's extremely interesting at the end to put them sort of together. Analyzing large population cohorts presents significant challenges, as we have learned. And researchers frequently highlight the key requirements. I very often talk to principal investigators and ask them, what are your biggest challenges, or what is it that you would require when doing a large cohort study? Number one, it's a robust technology that delivers consistent, reproducible results over time. Second, the most comprehensive coverage of analytes and biological pathways. Third, the full ownership of their data and ensuring that it can be made publicly available. It can be compared with other cohorts and used to produce meaningful results. Number four, a scalable solution capable of handling thousands to hundreds of thousands of samples, or in other words, from 5,000 to 500,000. Number 5, a fully decentralized approach, as we have seen also in the first and in the second talk, or as I like to call it, democratizing metabolomics. And in this slide, you see that how we from biocrates sort of set the gold standard in metabolomics with kits that deliver absolute quantitative data, standardized workflows, and robust validation. Through international ring trials, as mentioned, across multiple labs and MS platforms, our kits show excellent interlaboratory precision, ensuring reliable and reproducible data that can be compared and pooled globally across different platforms and instruments. And if you're interested in that, I'm happy to share the original publications with you. As highlighted in the previous slide, international ring trials have proven the reliability of our kits. As one example, the MXPIC-1500 that we've learned from Dr. Bach offers indeed an unmatched precision, quantifying several hundreds of metabolites, but with a very small sample volume of 10 microliters. And this trial that you see here, the ring trial, is led by Professor Jerzy Adamski and Gabi Kastenmüller from the Helmholtz Center in Munich, and we see the involved institutions that took part in this ring trial. And the outcomes of our ring trials, so to say, speak volumes, because the interlaboratory reproducibility is demonstrated by a CV of up to less than 15%. And that is based on completely different instruments, different mass spec instruments, because you know from your experience, if you analyze the same samples on a different instrument, you might get different results. And this is exactly that we would like to overcome. And it's actually more than statistics. It proves the lab-to-lab comparability, and it enables data pooling from multiple laboratories. You might remember this slide from your early days when studying biochemistry or any biology-related field. Today, we have actually advanced our technology to cover up to 1,500 quantitative markers, spanning key metabolic pathways such as the energy metabolism, lipid metabolism, and amino acids as well, just to name a few. And interestingly, also linking to the microbiome, what we have learned in the previous talk. This provides the comprehensive data essential for translational research. And very notably, we offer tools that enable the integration of metabolomics with other omics, allowing for a more holistic understanding of health and disease mechanisms. And that I will show you in the next slide, is that there are plenty of tools that we can discuss with you to support, first of all, the so-called fair data compliance. I'm not going in too much detail, but it's about the openness of your data, which is frequently requested by journals that you would like to submit your manuscript to. So it's complemented by a robust reference database, which is a quite unique reference database that we have here, the so-called QMDB. And also another tool that you might like is the sums and ratio tool called MetaboIndicator, which enhances the clinical relevance of the data. And in addition, we provide data compatible with various open source tools for pathway analysis, which is nice. So you don't have to use our tool if you like. You can additionally use our data to submit it to other software tools to do your pathway analysis and statistics. It's all compatible. And another point I would like to make here is that the methods that we use are following the FDA and EMA pre-validated analytical methods. And it's actually streamlined the transition toward so-called LDTs, so laboratory developed testings, or the so-called IVDs, so in vitro diagnostics, which will be facilitating the regulatory approval processes. I think that is an important step that is always a hurdle, but we have solved that problem already in advance. And this slide here is related to the, if you remember, the 5 points that I've made about the requirements relating to scalability. So we have a technology which is not only scalable, but it's also transformative. So by enabling a decentralized workflow and what I said as democratizing the access to advanced metabolomics, we have so far empowered more than 100 core facilities worldwide to contribute to global data networks. And this scalability ensures that even small laboratories can engage in quite groundbreaking research, and with comparable and poolable data, different instruments and labs, they can collaboratively work on the same the project, the cohort in particular. And I should also mention here that supported by the Andrew Plus automation solution in partnership with Waters Corporation, we're very proud that we can now be ready to scale up from 5,000 to 50,000 or ultimately to 500,000 or even more, and efficiently and seamlessly. So I would like to conclude here with a sentence that broad profiling is nowadays no longer a privilege of untargeted approaches. With our next generation targeted metabolomics kits, we can now generate up to 1,500 high quality metabolic data points designed to meet the precise needs of large cohorts and biobanks. Our solution offers standardization, quality control, fully quantitation, and reproducibility. And in addition, as I said, it's decentralized, it's scalable, and it ensures that users maintain the full control over their data and workflows. And in this slide, I would like to give you, before we finally conclude, some valuable resources that you might find interesting. So if you see here on the left side, there's a white paper on metabolomics in population health, which is a comprehensive guide to leveraging metabolomics for large-scale health studies. But I would also highlight another white paper by my dear colleague Alice Limonciel on the clinical impact of metabolomics. It's called the Complex Chronic Diseases Have a Common Origin. It gives you more insights into how metabolomics is shaping clinical research and personalized medicine. Please do also visit our website on epidemiology and biobanking. If you look into the application areas, there you can explore how the biocrates solutions can support large-scale cohort studies and biobanking initiatives. And I should also mention, which is a very nice one, is our video channel. It's actually a treasury of knowledge, a curated collection of talks from the world's leading experts on biobanking and cohort studies. It's really worth listening to them. Not all of them have used the biocrates technology. We've simply found them extremely interesting and wanted to have their views, and you can watch them obviously for free. And last but not least, if you're interested in a comprehensive literature list on cohort studies, you are always, you can feel free to send me a request. And I hope that these resources can be, they're designed to help you to deepen your understanding and make the most of metabolomics in your work. And with this, I would like to thank you for joining today's webinar. I hope you found it insightful, but also enjoyable.